Hi, you're watching DNA Today, a multi-award-winning genetics podcast where we explore everything to do with genetics, from CRISPR to rare diseases to new research. We have won the Science and Medicine Podcast Award for many years now. We have hundreds of episodes, and we really hope you enjoy these conversations where we dive into so many genetic concepts. I'm Kira Deneen, a certified genetic counselor and your host. In this episode, we're focused on the interpretation side of reporting and discussing how important the human touch element is to this process. Joining me for this discussion are two experts from Blueprint Genetics. We have Meena Mahey Kumar, who is the Senior Director of Clinical Genomics and supports the Clinical Genomics Services team at Blueprint Genetics. Meena has worked in pediatric, adult, and prenatal genetics before working in commercial genetics laboratories, leading to sales and marketing and clinical genomics service initiatives. Laura Fuqua is a genomic variant scientist for Blueprint Genetics. As part of the global clinical interpretation team, she analyzes variants for their pathogenicity in human diseases and collaborates across Blueprint and the parent company Quest to build genomic services. So, Meena and Laura, thank you so much for coming on. I'm very excited to be talking about this human touch element to interpreting genetic data because, in the age of AI, which I know we'll get into, this is really important and we don't want to lose this. So I thought I'd start our conversation talking about what does it mean to have the human touch in clinical interpretation? Because people might hear this and be like, that sounds like a very abstract idea. What do you mean by this?
It's such a pleasure to be on your show. Thank you so much, Kira, for having us. So that's such a great question. What does it really mean? Well, we have thousands and thousands of variants that could come up when someone orders a genetic test for a patient, and depending on what kind of genetic test it really is. But we're not really, when we're talking about the human touch, we're not really relying only on the algorithms that we use, the AI that's used, the structured data to spit out that list of variants. Rather, we need to use that human touch to complete the picture with trained intuition and really interrogate that presented data against what we know about that individual, what that patient's clinical and medical history is like. When we're thinking about the human touch, we're going through the variant interpretation process, the analysis, and while we we do use the ACMG and the AMP guidelines for variant classification, we still need to question every step of the way. Does this variant fit? Why does it not fit? What is the missing data? There's only so much you can get when you just have the data there, but how do you connect all those dots for that one individual patient and find that needle in a haystack, truly find that needle in a haystack? And what's exciting is all the humans that work on the variant analysis and interpretation are really honed into their trade, and they've gotten all the training. They still, participate in so many things. We have PhD geneticists, we have really well-trained genetic counselors and research scientists in analysis that work on the whole process of analysis and interpretation.
It's a lot of information to sort through, depending on what test you're doing, and a lot of things in our field is shifting over to tests that give us much more data. So there's so much more to sort through. So Meena, as you said, I really want to pull out a gem that you said, that we need to know what we're looking for in some sense. Like, what is the context of doing this test? What were the clinical findings that give us a hint of where to start looking, and how to process the information of, okay, this is like a variant that we've identified, but you have to see it within this full picture. And it's very hard, I would think, for computers to be able to take all of those pieces in. How are they going to match someone that has, say, a PhD in genetics or a master's in genetics, like us genetic counselors? So that's an important piece. There's a lot of pieces to that, but it is a really important piece. That makes me think, we have to strike a balance between the technical aspects of the genetic data and that computer science side, bioinformatics, huge data sets nowadays, but then balancing that with the human expertise needed for that accurate interpretation, because the interpretation part at this point in 2023, I would say we still need and will need a human to be part of that process for the foreseeable future. Maybe people listening in like 2035 are going to hear this and say, "Wow, that's different." But yeah, what are your thoughts on that, of striking the balance between the technical aspects and the human expertise side?
Yes, I can speak to that. And again, Kira, thank you for having us.
Yeah, definitely. Yeah, I want to hear what you have to say, Laura.
So I can echo what you said about things always changing, and that's how I see clinical interpretation. It's never static, and there's, like you said, there's an exponentially growing body of knowledge and tools that are available. So I like to think of it as an organism. It's always growing and changing. And I can speak to, at Blueprint, we do have that balance between the technical expertise and tools that are needed, but also the human expertise. And that is because both of them together give us the best possible interpretation. And that is much better, a much more accurate interpretation than relying solely on computational predictions. So I can speak to two of our technical aspects and also to our human expertise. So at Blueprint, we have carefully and thoughtfully designed algorithms that have been developed in-house by our teams of bioinformaticians and clinical geneticists that we continually refine and improve them. We serve over 70 countries with 14 different medical specialties, so we have a continuously expanding knowledge base, database that also has a higher representation of diverse patient populations. As far as our human expertise, Amina just spoke to this, and I can expand a little more, is we have many team members who are active in the genetics community. So we have many team members that participate in the ClinGen Consortium, in the NSGC special interest groups. We have many team members who maintain active board certification. We're always open to scientific collaboration. And this is kind of exciting. I wanted to share this, that one of our board certified directors, she is a part of the ACMG sequence variant working group. And they are working on a draft of version 4 of the ACMG AMP sequence variant interpretation guidelines. So that was just presented to the ClinGen Consortium last week. It's a draft in progress, but it really highlights and shows that we have team members who are out there on the front lines in the community, building the tools, and at the using their expertise to improve our community.
Yeah, that's so important, and how cool of them to be able to be part of this working group and be changing things in real time. And, we say it all the time on the show, genetics changes very fast, and it's exciting to be a part of that change, and all the ways that, your teammates are part of that. And we've touched on a little bit, I wanted to come back to artificial intelligence, AI. I mean, I think that's, like, if I had to say the word of the year, I think is AI for 2023.
Definitely.
And, I started even changing my podcast workflow of just how we're integrating AI into it. And with ChatGPT launched, it was November 2022. I didn't really start hearing about it, I would say, until the new year. But, with all of that makes me think, well, how is AI technology impacting the role of human interpretation in genetic reporting? Like how is that all coming together? Like, what are, I don't know, like maybe you guys have used this, or maybe are thinking about using it at Blueprint in the near future, of different strategies to take place to still ensure that human touch aspect is included and a part of the process, and AI as a tool, and it's not replacing work. Because I think that's a topic that gets brought up in a variety of areas. I mean, even like the writer's strike and different things that we see outside of genetics. But, we're focused on genetics here, but, I'm just curious to hear from either of you on if there's been talk about including AI. Have you already done that? That could be a whole episode in itself. Stay tuned, listeners. I'm sure I'll have some.
Definitely. Laura, do you want to take that? Because we talked a little bit about how, we use AI, and we definitely do. But I'm going to let Laura take this, and then maybe I can add at the end.
Yeah, I can share answer your question, an overview, and how we have applied these tools at Blueprint. So, as we all know, as genetic counselors, the AI technology is fantastic and has a significant impact on enhancing efficiency and accuracy. It can handle this vast amount of genomic data that NGS creates for us. But it's the nuanced interpretation that, especially that in borderline or uncertain cases, that still rely on the human expertise. So think about the decisions on what to report, how to communicate potentially sensitive information. This still requires, and will always require, human judgment. And when we think about scientific discovery, AI uses the model of what is already known. So looking at new patterns and connections outside of what is known is a limitation of AI. So the way that the AI is going to change the landscape of our ecosystem is that I see it freeing up the routine tasks, like going through it and sifting through all of the data, and it's going to free up interpretation teams for those complex cases and for discovery. And we like to think about a genetic report. It's not just about the genes, it's about the individual. And we think that we will always need those human experts who can integrate those molecular and clinical findings, take into account the family history and other factors when deciding on what to report. So humans will continue to play a pivotal role. As to what Blueprint has is we do have a proprietary tool that our data science team has created, and it goes and sifts through the data, and it kind of gives us help by predicting the pathogenicity of a sequence variant, whether it's pathogenic or disease causing or more on the other end of the spectrum or benign. It's a machine learning classifier. So it's already a nice example of using AI to do that work, so then the interpretation team can sort of work at a higher level with that data that AI generates.
Yeah, that's so helpful. It's interesting with AI and being able to use that as a tool to speed up your workflow so that you can handle more projects and be able to sift through data quicker. So really using it as a tool, like we said, but I think it's also important to look at how are we training these AI programs and whatever the term we want to use is, because if we're giving it bad data, it's not going to do a good job at sifting through the data you're giving it, right? So there's this phrase I've heard a lot in terms of AI of, good data in, good data out, bad data in, bad data out. So I think when companies like yours, I'm sure, were designing a lot of these AI modules of, okay, we got to figure out how are we training it? And for companies, like yours that have patients from so many different places in the world, that also I imagine helps out with data being diverse and not being from all of the same area of the world, ethnicity wise. So I think that's also really helpful and a topic that we talk about a lot on the show, because I think it's very important that, in 2023 and as we move forward, that we are keeping that at the forefront of preventing, disparities in healthcare. So I think that's great. And just, that's one worry of mine, I think, with AI. Lots of excitement, lots of worries, just like we talk about a lot with genetic technologies. But it's really cool just to hear how you guys use it in terms of looking at variants and helping to sort out and saying, okay, we think this one could be pathogenic. Maybe this one is likely benign, and kind of helping to start that process. We've been talking about a lot of this in terms of general concepts and, sorting through data, AI, keeping that human touch on, looking at the clinical case. But I want to get a little bit deeper in terms of talking about how the human touch and interpretation contributes directly in impacting patient care. Because at the end of the day, the reason we're all sitting here recording this episode is because it comes back to learning about genetics so that we can help people's lives. So that is everybody's goal. So how is the human touch important in making sure that that is still our goal and that's still the priority? I don't know. I don't know if either of you kind of have initial thoughts on that.
Yeah, I definitely think I can maybe speak to that one, is just with, this whole, all the things that are out there now, we still, like you said, need humans, because we need to critically think We need to make those judgment calls. Like you said, there could be bad data out there. Doing that critical thinking is so important. And that critical thinking that really leads to that needle in the haystack answer. Searching for that answer, leaving no stone unturned, is going to require a human sifting through the data that we require, that we've acquired from the different algorithms that we've used, the AI that was used, all the tools. And it really helps us, if we do find the answer, to advance possibly the clinical management of a patient, right? So that's a big impact. And of course, we've all been there where GCs just finding an answer for a patient, even if it doesn't change anything for them in their clinical management, it still changes the world for them and how they perceive everything, how they think about themselves, how they identify with themselves, and they can go on and continue on in their journey. And it doesn't stop there when you have a clinical report, right? For a patient, they take that with them for a long time. It's a story about how we've classified a variant. There's all the evidence that's there. At Blueprint Genetics, we love to put all the links to the PMID articles, so you can just click them, and a care provider can look into why we've classified a variant by looking at the same research that we've looked at, looking at the same OMIM numbers that we've looked at. So it's just the beginning. Delivery of the results is just a beginning. We have the results, and we still don't end there. We're there as collaborators at the lab. We have genetic counselors who can answer the phone if you have questions, or if there's questions later, we feel like something's missing or something new comes up in a patient's phenotype. We can still go back to that clinical report, go back to that interpretation, go back to the data and do more with it. And that still requires the human touch, if you want to, think about it that way as well. So I really think that there is always going to be the need for that human touch to impact patient care throughout the whole process.
Great points, Meena. And I think, too, I want to pull out of that is that transparency is so important, of saying, this is the reason we classified this variant as we did. This is why we're calling it pathogenic. This is why we're calling it a variant of unknown significance or likely benign. And these are the studies that are supporting that. So people can do their own research and see, okay, I see where you're pulling this from. Or they could come back at you. I'm sure you've had providers do this and say, well, have you seen this study? I don't know. This conflicts.
Yes.
And then you work together. That's the beautiful part of the collaboration, that it's not, oh, I found you wrong or something. It's, we're always learning. So we're always adding more, and, more good data, the better, to come up with this answer. And that situations do change, as you said, Meena, that we can say, well, this is the symptoms so far, and then we could have a regression of symptoms, of kind of losing milestones that maybe that baby or child had before. So I think it's really important that, in those cases, we could go back to the raw data and say, okay, well, now let's sift through it with this lens, which, I don't know, that's going to be hard for a computer to do. I'm thinking 100%. Of diving, so diving even deeper, if we can get into maybe an example of a situation, or, different situations kind of clumped together, of where the human touch did play a crucial role in actually interpreting and reporting out that genetic information, of, maybe using some of this technology, but along with an expert that's looking at it that is human to give that information. Is there a case that comes to mind for you guys?
Yes.
Oh, we have so many. But yes,
You're
I think, Laura, go ahead.
Yeah, how long do you have?
Yeah.
And I love this question because it really highlights the why of what we do. So I have two cases, and I hope I have time to do both. So the first case, we had an individual who was reported as unaffected, and the client or customer or ordering provider had ordered targeted testing. So that means that this variant was previously identified in one of their relatives. And we identified a diagnostic result in a gene called MYH9. It's related to thrombocytopenia. However, the NGS data required more analysis. It required the clinical bioinformatics team and our interpretation team to really look carefully at the quality of the data, because the data supports that there was low-level mosaicism present in the specimen that was provided to us for testing. So having that technology there to really hone in and give the most accurate interpretation to the result. And prenatal clinicians like yourself, Kira, I think you can really appreciate that, how this directly changes the medical management of this individual. So back up here, I left out the really important part, is that we had clinical information from the client and it was invaluable. In this case, it's a pregnant patient. So having this result in this gene, Carrie, you can appreciate as a prenatal clinician that this directly changes the medical management.
Yes.
Now the doctors can think about ordering platelet counts. They can make a delivery plan for management of the delivery in case they need rapid blood access at delivery because of the chances of postpartum hemorrhaging or delivery complications for the neonate. So I love that example because it has the technology, the collaboration with the clinician or the ordering provider, and then what that actually means for this person and their pregnancy.
Yeah,
And
definitely.
I just want to double click. Sorry, Kira. I just wanted to double click because this low level mosaicism piece isn't something that you're going to quickly be able to, that AI is going to be able to spit out and talk to you about, right? It's really our bioinformaticians that look at that and analyze it and flag it for the team, and the team can go back to the data and look at the reads and look at the mosaic reads there and determine whether or not they can confidently call out a mosaic result. That really exemplifies human touch here for sure.
It
Sorry,
really
thanks,
does.
Laura. That was a beautiful example.
Yes.
Yeah, definitely. I think mosaicism is just such an interesting genetic concept. It's one that really fascinates me. And for people that aren't familiar with this, it's when a person can have two different cell lines. So one cell line could be normal cells and one cell line could have a difference. So sometimes we see this in people that have Down syndrome. Some of their cells have that extra chromosome 21, some do not. And you can have a mix of the two. So just interesting that it makes sense to me if there's only a tiny, tiny percent of those cells that are different, that other cell line, the computer's probably just going to think, oh, this is just, background noise in the data. It's not actually important, where a human looks at it and starts looking at ratios and says, well, we keep seeing it throughout the data at this certain level. We got to look further into that. So yeah, I love that case. And I know we've said NGS a couple times, next gen sequencing, for people also that aren't familiar with that, which 95% of our audience is, very much so. It's part of our vocabulary. But Laura, you said you had one other case.
Yes. So this highlights one of our interpretation team members really going that extra mile and using their instinct about a case. So our pipeline called two variants in this gene called ATAD3A. So this is related to a syndromic condition. And the information that we had, the clinical information, was that it was an infant with congenital cataracts, but no syndromic presentation. And this person just felt that, going that extra mile, just continually digging into the literature search to find anything to give them more evidence and how to interpret this result. And they found a recent publication describing this same gene, the same variant, and a patient with really similar features to the one that we were testing that also had emerging phenotype. And why that's so important, I think, for the patient, first of all, like Meena said, it gives them an explanation, an answer. But also, I think it's really important for the human expertise, experts in interpretation to go that extra mile, because with NGS technology and more people electing it, we're going to find out more about emerging phenotypes. Especially for these syndromic conditions, things that are not well known, that may only be in a recently published paper. So I think that was a great example of someone just using their expertise and just not giving up.
Yeah, and importance, like case studies are so important because sometimes it just takes one case study to say, well, now we have a little bit of data to support this. So yeah, that's really cool to find a case study that really much lines up with the current patient that you were helping.
Which is such an exciting feeling. I think that, when you're working with these cases, it just is like such a triumphant moment. And then you feel like you're making such a big impact. And I think that's what really inspires the team to do what they do as well.
Yeah, it definitely does because when you're able to provide a person or a caregiver a specific diagnosis or more information, then they can go out and find other people that are in that community and they can have that sense of community, which is really important too. We've highlighted a lot on the show of just that is often a silver lining to a diagnosis and just having people that really understand where you're coming from. But yeah, that's a really interesting case. So we've talked about how this has worked with a couple different cases, and the process can be different for different conditions or indications. How do you guys process this differently when you do have indications that are wildly different? I mean, sometimes you have things coming from the prenatal area, like we've been talking about a bit. Sometimes it's in peds, sometimes it's in cancer. Is there a difference in, just the types of genetic tests being ordered and how you start processing this in terms of breaking down that information?
I can start with an answer for
Sorry,
this one.
Laura. Yeah, please.
Um, and again, I have two examples. So, yes, our clinical interpretation team, we do different things for different conditions. And that goes back to our carefully designed algorithms. And to expand on that, the first example I have is the RPGR gene, which is related to X-linked retinitis pigmentosa. RP affects about 1 in 3,000 individuals. And so there's a region within the RPGR gene, it's called OR15, that has limitations with NGS technology because of the biochemistry of the region. It's highly repetitive and purine rich. On the other hand, that same region is also a hotspot for disease-causing variants. So most of the disease-causing variants within RPGR are in that OR15 region. So Blueprint recognizes the importance of this region and developed a novel test to increase the coverage of the region. But we take it a step further in that our clinical bioinformatics team and our interpretation team also look extra closely at the sequencing data in this region to solve these cases because of the limitations of NGS technology, looking for these smaller deletions. So that's for RPGR. There's another example, also an ophthalmology example, but we also know that these mobile insertion elements have limitations with NGS. And so there's an ALU insertion in the MAC gene. It's a well-characterized founder variant that is related to autosomal recessive retinitis pigmentosa in a certain population. So how we solve this is that, again, our bioinformatics team developed an algorithm or a tool to have increased coverage to identify these ALU insertions, but we also take it a step further in that we look at the data and we check for drops in coverage just to ensure that we do our very best to make sure that we check everything for accuracy, especially when the indication is unexplained retinitis pigmentosa and there's no other possible genetic explanation that we identify.
I think Laura brings up such a good point because she talked about some, really special cases in the ophthalmology disease category that we have. And at Blueprint, we have 14 different disease categories. And when you think about it and you think about how the interpretation process differs, we talked about those two genes and what's nuanced in those two genes and what we need to pay attention to. It really does mean that across all these different disease categories, you've got to develop that expertise. And going back to the human touch and the human element of that, It's making sure that our team is educated on their disease category and specializing in certain disease categories. You can imagine it's really difficult to be an expert on all of them. So we really have teams of geneticists and genetic counselors that focus in on a few, and that's what they're refining their skills on. That's what they're building their knowledge around and interacting with people in that same community and that same expertise. And it's just, I've watched the team develop over time. So I've been here since 2016 at the beginning of that year. And we started off as a cardiogenetics lab. And as we added all those disease categories.
That's the origin, yes.
That's our origin story. And it's so like academically rooted. But watching how the team has grown and how they've developed their skills and how they organize around that and how they interact with clinical consultants as well in case there are questions that they can't answer and need to link back to the, clinical phenotype. It's become such an art, right? And that's where that art and science really meet up. And I love watching that. And I really think that's also part of the human touch, that constant education that you need to have, that constant community that you need to rely on in terms of developing the expertise in analyzing and interpreting different types of genetic tests, right? The panels, even whole exome is a bit different than doing the panels versus the disease categories versus the single genes. So just to, kind of sum that up in how we do it.
Yeah, and you guys have done such a great job of expanding out, your Blueprint genetics community and education to our listeners, of kind of letting them get an inside look at how you guys add this human touch element to your processing and interpreting of all of this genetic data, and just working together. That's something that is always going to be very advantageous, that we all have our different specialties and skills, and to be able to work together, of not just one human looking at it, but many humans looking at it and being able to collaborate on that. You're just going to get better reports, I think, of, the results is one aspect, but having that part where you're breaking it down for the provider that's ordering it, because even if it is a genetic counselor, I very much appreciate that, because here's my rundown, right? But especially for people that are not trained in genetics, that are ordering some of this tests, and they may have just heard like, oh, okay, did a little research and ordered it, but don't really fully understand the results, that we really need those reports. And I think that's why it's so important to be looking at labs and looking at sample reports for, younger GCs out there that are kind of at that point of, okay, I need to start choosing what labs I'm ordering from. I think in general, that's something that I made sure to ask when I was, just first meeting with companies and figuring out, how to start approaching those relationships and everything. But thank you, Meena and Laura. This was really cool. It was really interesting just to dive into this topic, but then be able to touch on so many specialties while talking about it. What did we span? We talked about prenatal, ophthalmology. We got some adult conditions in there. So, and that's just off the top of my head. The listeners are probably like, here's forgetting this one. But it was just lovely to have you guys on and nerd out about AI is awesome and scary all at the same time, and we need humans to keep it in check.
It was so great to be here. And I wanted to double click on a little bit what you said about patients and providers themselves. That's really where all the clinical information comes in, that ties everything together to get that best result. So we invite all the information and collaboration from them as well.
Yes, and I'm so excited to see you guys next week at NSGC. It's going to be fantastic. Really excited to be in Chicago, get my deep dish, get all my genetic nerd talk on in person. So very pumped to see you both there next week.
I'll be looking for you.
Definitely. Thanks. It'll be fun to see you, Kira. Thank you so much.
Definitely. Thanks, guys. Thanks for watching DNA Today. To access all of our episodes, head over to dnatoday.com. We also have a lot of bonus content on there that you can enjoy. If you have any questions, comments, suggestions, guest pitches, you name it, send them in to info@dnatoday.com. We'd also really appreciate if you could take a moment to rate and review the podcast on Apple, Spotify, wherever you listen to the show. It really helps more nerds like yourself find the show. Also, if you like giveaways and other ways to connect with us, I recommend following us on social. We're at DNA Today Podcast. We also have a Patreon if you want to be the most level involved in the show. That's also at DNA Today Podcast. Thank you so much for listening and watching. Join us next time to learn and discover new advances in the world of genetics.